Welcome to Biotanemy.
Bioremediation. PCBs, due to their hydrophobic nature, partition from soil organic matter into the lipid membrane of dihalococoides myocardiae, where they interact with membrane-associated reductive dehalogenase, a chlorinoid or vitamin B2-dependent enzyme that catalyzes reductive dechlorination. A specific carbon-chlorine bond is oriented toward the catalytic center. The enzyme environment stabilizes the substrate for reduction. The electrons flow through the membrane electron carriers into the chlorinoid cofactor. The reduced cofactor transfers electrons to the PCB. This weakens the carbon-chlorine bond and causes heterolytic cleavage. The PCB now have one chlorine less, making it slightly less hydrophobic and more bioavailable. This PCB congener passively diffuses out of the membrane due to reduced hydrophobicity and concentration gradients, re-entering the environment where further degradation can occur. This PCB now acts as a carbon source for organisms like Burkholderia xenovorans Lb400 and Pseudomonas putida. It diffuses into their membrane and reaches the cytoplasmic side or enzyme-accessible region. The biphenyl dioxygenase or BPH enzyme complex binds the biphenyl structure and uses oxygen, NADH, and ferrous ion to insert two oxygen atoms into one aromatic ring. Ferrous activates oxygen to produce reactive oxygen species. Both oxygen atoms are inserted into adjacent carbons of the ring. Double bonds rearrange, and the ring loses aromaticity to produce cis-dihydrodiol PCB. Next, the enzyme dihydrodiol dehydrogenase, BPHB, will then remove two hydrogens from cis-dihydrodiol to form catechol-like dihydroxylated PCB and restore aromaticity. In the next step, the enzyme 2,3-dihydroxybiphenyl dioxygenase, BPHC, will add oxygen again to break the aromatic ring through metacleavage. This step will destroy the PCB ring structure and convert it into a linear compound. The enzyme hydrolase or BPHD will split the linear intermediate into chlorobenzoic acid or CBA and smaller aliphatic compounds. This is still partially chlorinated, so it undergoes dechlorination hydroxylation via the benzoate or catechol pathways. In that process, CBA is enzymatically converted into chlorobenzoyl-CoA by a CoA ligase with the expense of 1 ATP, by the enzyme chlorobenzoate-CoA ligase. The enzyme chlorobenzoyl-CoA dioxygenase will convert chlorobenzoyl-CoA to dihydrodiol-chlorobenzoyl-CoA and NAD+ with the help of oxygen, NADH, and H+. Dihydrodiol-chlorobenzoyl-CoA and NAD formed will be converted to dihydroxylated chlorobenzoyl-CoA and NADH by dehydrogenase. The enzyme dioxygenase will insert oxygen between hydroxylated carbons to open the aromatic ring to produce a linear dicarboxyl-CoA intermediate. The enzyme dehalogenase facilitates the hydrolytic removal of chlorine from the intermediate. Following ring cleavage, the resulting cis-mucinate undergoes isomerization catalyzed by mucinate isomerase, which rearranges the double bonds and cyclizes the linear molecule to form muconolactone. Subsequently, muconolactone is converted into beta-ketoadipate enol-lactone by the action of muconolactone isomerase, which facilitates an internal rearrangement of double bonds and functional groups, preparing the molecule for further processing. The beta-ketoadipate enol-lactone is then hydrolyzed by enol-lactone hydrolase, in the presence of water, resulting in the formation of beta-ketoadipate, a linear dicarboxylic acid containing a beta-keto functional group. Finally, beta-ketoadipate is activated to beta-ketoadidyl-CoA through a CoA transfer reaction catalyzed by beta-ketoadipate, succinyl-CoA transferase, in which succinyl-CoA donates the CoA moiety, producing beta-ketoadidyl-CoA and succinate as products.
Phyolytic cleavage occurs with the help of beta-ketoadidyl-CoA thiolase will break the beta-ketoadidyl-CoA at the beta-keto position using CoA as a nucleophile, splitting it into acetyl-CoA and succinyl-CoA and enters the TCA cycle.
That's all for today's video. Thank you for listening. Hope all of you understand the concept, and for more queries and proposal, please comment down. See you all in a new topic.